Welcome students to another video, this time on translation. We continue our journey in chapter 7, focusing now on translation, the process by which messenger RNA is converted into polypeptides. Translation requires the interplay of 3 different types of RNA and a bunch of different proteins: messenger RNA, transfer RNA, and ribosomal RNA. In Module 1, we look at transfer RNA. During this process, we should be able to understand and explain the composition of transfer RNAs themselves, and also describe how transfer RNA molecules are loaded with amino acids in the cytoplasm, a process known as charging. The following 4 concepts will be discussed in this module, namely the composition of transfer RNA, the enzymes involved in loading the amino acids onto the transfer RNA molecules themselves, and the concepts of a codon and an anticodon in the process of translation. In our PowerPoints and our textbook, transfer RNA molecules are represented on the pages and figures in many different formats, but they all represent the same general structure. Illustrated here in panel A are 2 alternative ways in which the transfer RNA molecule is depicted in 2 dimensions. Very importantly, most of these molecules are between 70 and 90 nucleotides in length. They're made of RNA, therefore they have a 3' and a 5' end, and they contain uracil instead of thymine. They do form small regions of secondary structure as indicated here by the red lines joining the 2 halves of the molecule. Loops are formed in various regions, and the number of loops is always 3. So this loop in yellow, the loop in red, and the loop in blue are particularly important in dictating the overall structure of this molecule. Hence, the shape of a clover leaf is obtained, and the use of that is instrumental in the terminology used to describe these structures. The 3' end is called the acceptor stem because that will accept an amino acid, and the other side, the blue region, known as the anticodon loop because it carries these 3 very important nucleotides which are instrumental in making contact with messenger RNA within the ribosome. Panel B and panel C depict the transfer RNA molecule in a 3-dimensional orientation. The difference between panel B and C is that the molecule is just rotated 90 degrees around. This folding causes the amino acid attachment site to be brought down to one side, and the anticodon loop is organized facing south. Panel D simply shows us a shorthand version of how a transfer RNA molecule will be represented in subsequent slides. Panel E simply shows us the linear sequence of RNA nucleotides in one type of transfer RNA molecule. Transfer RNAs are generally considered as the go-betweens matching the genetic code to the amino acid code. Transfer RNAs, they collect from the cytoplasm the amino acids dictated by their individual forms, and they deliver those amino acids to the ribosome so that they can be matched up with messenger RNA. We're going to learn this process shortly in module number 2. Amino acids derived from food or manufactured by enzymes and biological pathways in the cytoplasm are free-floating entities. They may be coupled to transfer RNA molecules based on the presence of specific enzymes within the cytoplasm. It's very important to realize that there is one enzyme for each amino acid which needs to be coupled to a transfer RNA molecule. As far as we are concerned at this time, there are 20 such enzymes in the cytoplasm of all living cells. The process involves 3 steps in all cases. Amino acids are first combined with high-energy phosphate groups to form an adenylated amino acid, which contains a high-energy molecule. Then those high-energy amino acids are then attached to the 3' end of the transfer RNA transferring the energy to that molecule. This is a ester bond between the oxygen and the carbon sugar of the transfer RNA. The entire process takes place in the active sites of the enzyme. The enzyme is depicted in green in this case. It has two binding sites, one for the amino acid and a second binding site for a particular transfer RNA set. So this amino acid will only be bonded to a particular type of transfer RNA. None of the other transfer RNAs, which are designed by nature to carry alternative amino acids, can fit within this binding site. So once again, the shape and charge of the molecules, the chemistry, has to be compatible in order for a chemical reaction to proceed. If the amino acid and the appropriate transfer RNA are able to bind to the active sites of the enzyme, then the reaction continues and it results in the loading of the amino acid onto the end of the transfer RNA, a process known as charging of transfer RNAs. These charged transfer RNAs are then released into the cytoplasm to become freely available as a pool of usable transfer RNA molecules. The right side of this figure takes place within the body of a ribosome. Here, the appropriate location of a messenger RNA codon is placed into a binding site which tries to match up the anticodon on the transfer RNA to the codon on the messenger RNA. So this is a very critical location that takes place inside the binding sites of ribosomes. If the appropriate transfer RNA is able to bind to the codon, then the amino acid is left behind within the ribosomal body. If the transfer RNA is not complementary to the codon, then it is ejected without the amino acid being kept by the ribosome. That concludes this simple module where we have explained the composition of transfer RNA, and you should now be able to describe in detail how transfer RNA molecules are charged with their corresponding amino acids. Module 2 explains the structure of ribosomes, which are key to the process of translation. By the time we finish this module, we should be able to describe the structure of ribosomes and then also compare and contrast prokaryotic ribosomes with eukaryotic ribosomes, illustrating any differences between the two. These 2 bullet points from the word bank from Chapter 7 will be tackled in this module. If we confine our analysis to eukaryotic ribosomes initially, we can see that there are 2 locations in which ribosomes can be found producing polypeptides. The first location is free in the cytoplasm. Free ribosomes release their polypeptides directly into the cytosol. Bound ribosomes, on the other hand, are only doing translation on the surface of rough endoplasmic reticuli, therefore delivering their polypeptides not into the cytosol, but either into the membrane of the endoplasmic reticulum or the lumen of the endoplasmic reticulum. This is a very important distinction to be learnt. It is virtually impossible for proteins which are made in the cytosol to end up in the lumen of the ER, and conversely, proteins which are made in the lumen of the ER are very difficult to find in the cytosol. Therefore, manufacturing the polypeptide in the right location is key to keeping it in that particular compartment. Ribosomes can be easily seen with the electron microscope. They are pretty large structures. They are composed of 2 subunits, a large subunit and a small subunit. The large subunit itself is 2.8 million Daltons in size. This massive protein complex is actually composed of 49 proteins and 3 RNA molecules. So once again, RNA has an important role in translation. Without the RNA, the functionality of the ribosome would not be apparent. The small subunit, which itself is just over 1.4 million Daltons, is also composed of a large number of proteins, 33 in eukaryotes, plus an important RNA molecule. The three-dimensional structure of a ribosome is shown in this particular slide. Viewed from different angles, one can see that the RNA molecules and the polypeptides come together in a very sophisticated arrangement. But what is apparent are the three locations upon the ribosome where most of the catalytic activity in producing polypeptides takes place. These are indicated by the red, the orange, and the yellow locations respectively in these illustrations. The E site is where the polypeptide chain exits, therefore the letter E. The A site is where the amino acids are purchased and brought into the ribosome by the transfer RNAs. And the P site is where the individual amino acids are combined together to form the polypeptide. So A for amino, P for polypeptide, E for exit. The final slide in this module illustrates the difference between ribosomes from eukaryotes and those from prokaryotes. Generally, the two are made up of the same subunits, the large and the small. However, the composition of each is different. The large subunit in bacteria is much smaller than the large subunit in eukaryotes. Not only does it possess fewer proteins than the corresponding eukaryotic subunit, it also possesses one less ribosomal RNA. The small subunit likewise has fewer proteins, but it does possess a similar sized ribosomal RNA. This is all that is required to satisfy Module 2. Students should now be able to describe the general structure of ribosomes and also compare and contrast eukaryotic with prokaryotic. Moving on to the final module in this sequence, that is the process of translation itself. The learning objectives here are to be able to describe how the structure of the ribosomes themselves makes translation possible. Also to describe the role played by the APE sites that we mentioned in the previous module. And finally, to describe the interactions between the messenger RNA, the transfer RNA molecules, the polypeptide chain, and the ribosome. This is the translation machine. Naturally, a large number of words from the word bank for Chapter 7 will be employed in describing this process. We begin by revisiting the assembled ribosome, the large subunit and the small subunit sitting together. They form within them three cavities or caverns. These caverns are very important in bringing together all the components for translation. The process is identical for eukaryotic and prokaryotic ribosomes. The order of these three sites must be learnt, so APE, or the word ape, is the order in which these sites are used during the manufacture of polypeptides. The assembled ribosome attaches to the messenger RNA in a particular orientation. So the 5' end of the messenger RNA is inserted into the small subunit of the ribosome, and then the ribosome moves along the messenger RNA until it finds the starting point for manufacturing the polypeptide. Once those codons are positioned appropriately, the large subunit attaches to the ribosome and starts the process of translation. In this and subsequent figures, the ribosome is going to be moving from left to right along the messenger RNA molecule. New transfer RNA molecules will be entering in the A site, then being shuttled along to the P site. If there is enough of a correspondence between the codon and the anticodon between the messenger RNA and the transfer RNA. Should there be a disconnect between the two, the transfer RNA will be ejected at this point, and a replacement transfer RNA will be brought in to start the whole process again. Since there are 20 different types of transfer RNA within the cytoplasm, there is 1 in 20 chance that the right transfer RNA will end up sitting in the A site. Regardless, that is how the system works, and it is very successful. Once the transfer RNA has moved along to the P site, then a polymerization event takes place in the formation of a polypeptide. So the amino acid belonging to that transfer RNA is then covalently linked to the preceding amino acid that was there previously as the polypeptide chain is growing. Once that has taken place, the ribosome once again scuttles along to the next codon. In this case, the transfer RNA is pushed over to the E site, and it is then injected to be recycled back to its enzyme to be reloaded with a fresh, brand new amino acid. The process cycles again and again until the messenger RNA reaches a termination point at which the process stops. The polypeptide is released, the messenger RNA is released, the 2 subunits are released. This slide and the next 3 show a sequence of events depicting the movement of the ribosome along the messenger RNA. So this is a replica of the previous slide. Once the correct amino acid has been delivered to the A site, the polypeptide chain is then broken from its transfer RNA and then at the same time relinked to the new amino acid, as indicated here between 3 and 4. This keeps the polypeptide chain from floating away and anchored to the ribosome until its future release. The next step is the movement of the large subunit of the ribosome relative to the small subunit. The large subunit moves across 1 codon to the right. At the same time, the small subunit holds on to the transfer RNAs, preventing their movement. In the process, the transfer RNAs are repositioned within the 3 caverns of the APE, APE, as indicated here. Finally, the small subunit ratchets along by a codon to catch up with the large subunit, recreating the APE sites, and the process now starts all over again until, as we mentioned earlier, the messenger RNA tells the ribosome that it's reached the end of the growing polypeptide chain. The manner in which messenger RNA is first attached to a ribosome is different between prokaryotes and eukaryotes, and this difference is very, very important. Let's start by looking at what happens in a eukaryote. In eukaryotic translation, the small subunit first attaches to the initiator transfer RNA, which in most cases is going to be the one carrying the amino acid methionine. It doesn't attach to the A site, rather it attaches directly to the P site. This primed small subunit then seeks out any messenger RNAs in its vicinity and attaches to them at their 5' end. Then it travels along the messenger RNA looking for the very first initiator codon, which is always going to be AUG. So AUG is the first codon at which the small subunit will come to a halt in the process of translation, as indicated on this slide. Once the small subunit has found the AUG site, the first one from the 5' end, then the large subunit is called in to form the structural ribosome. The ribosome then now has the E, the P, and the A sites automatically formed, and the process of elongation continues until the ribosome reaches the stop site somewhere further along the messenger RNA molecule. The process of initiation in prokaryotes is completely different. There, the ribosomes do not scan from the 5' end, rather they attach randomly to the messenger RNA along its length. They then scan both upstream and downstream looking for a start site, an AUG in this case too. Once they do find the AUG start site, then they proceed in the 5'3" direction in making a polypeptide. This fundamental difference between prokaryotes and eukaryotes makes it possible for prokaryotes to have multiple proteins encoded by a single messenger RNA molecule. We've seen this in the case of the lacopron. The lacopron has 3 individual polypeptides made from a single messenger RNA. Whenever a single messenger RNA encodes more than one protein, the term from genetics polycistronic is applied. So this is a polycistronic messenger RNA. This is not found in eukaryotes. In eukaryotes, there's a single AUG start site and a single polypeptide generated from that messenger RNA. Let's turn our attention now to how translation is terminated by ribosomes. The process of termination is relatively straightforward. When a particular stop codon, one of 3 stop codons, enters the A site from the messenger RNA of the ribosome, there is no corresponding transfer RNA. In its place, nature has a protein called a release factor. The release factor then matches up to the stop codon, enters the A site, and causes the ribosome to release the polypeptide chain, the messenger RNA, and its small and large subunits. So everything dissociates. This sequence of events is illustrated on this slide and on the next slide. The messenger RNA may now be available to be translated again by another ribosome, or it could be degraded by other factors within the cytoplasm. Indeed, the system is very capable of generating large numbers of polypeptides whenever the cell has a need for such. This slide demonstrates the existence of a polysome, a single messenger RNA which has upon it multiple ribosomes, all transcribing one after the other like a train. So a polysome is simply a structure consisting of a single messenger RNA with multiple ribosomes. This permits the cell to amplify the product from this single messenger RNA if such a need arises. Each ribosome still has to scan from the 5' end looking for the AUG site, as we discussed earlier. Once the ribosome assembles at the AUG site, it then moves along the messenger RNA. This allows another ribosome to attach to the 5' end, and so on. When the ribosomes get to the stop codon, they dissociate, releasing their polypeptide. Those ribosomes may then attach again to the 5' end and repeat the entire process. The structural differences between eukaryotic and prokaryotic ribosomes makes it possible for chemicals such as antibiotics to be designed to attach and block ribosomes from prokaryotes rather than ribosomes from eukaryotes. So if a person has an infection of prokaryotic cells like bacteria, then they can take antibiotics and not harm their own ribosomes, but block the ribosomes of bacteria, preventing them from translating their proteins. Let us conclude this video by looking at the genetic code. We spoke about codons and anticodons when it comes to messenger RNA and transfer RNA. What is a codon? A codon is simply a 3-nucleotide sequence which codes for a particular amino acid. The genetic code is more or less universal, with a few exceptions here and there. This code tells us which sequence of nucleotides on the messenger RNA correspond to which amino acid being added to the polypeptide chain. For instance, a GCA sequence written from 5' to 3' on the messenger RNA will add an alanine to the polypeptide chain. Since there are just 20 amino acids and 64 possibilities for the codons, nature has come up with a solution where multiple codons can code for the same amino acid. Before we look at those, let's look at the anomalies. There are, out of 64, 3 codons, UAA, UAG, and UGA, which, when present on the messenger RNA, result in the attraction of the release factor to stop translation. So those are known as the 3 stop codons. The other codon of note is the AUG codon. This codon, when present on messenger RNA, results in the initiation of polypeptide growth. It brings in the amino acid methionine. So in almost every case, the first amino acid added to any polypeptide is methionine. Now, this may be removed after the polypeptide is made, but regardless, the AUG codon is the start codon, and methionine is always initially added to that polypeptide. Some amino acids have more than one codon. In fact, the majority do. Some of them, as indicated here, for serine, for leucine, and for arginine, 6 codons each. This is known as the redundancy of the genetic code. You are strongly advised to read the colored pages within Chapter 7, which give a historical account of how the genetic code was deciphered using experimentation. This concludes our overview of translation. Thank you.